Okay, thank you for the kind introduction. Uh, Today, I will talk about some unfamiliar story to the nuclear medicine physician that is related with nanomedicine. But I think sooner or later that kinds of radio-labeled nanomedicine can help the development of nanomedicine or itself that can be used as the Theranostics. Uh, I think you may know that chemistry, that nano is the field of chemistry that word from the Greek word nanos, it means dwarf. So simply that nano is the size dimension. So 1 to the minus 9 is the nanometer size. And firstly, Nobel Prize winner Richard Feynman, he said that there's a plenty of room at the bottom. So that means we can magnify some material into the nano size that has some kinds of different phenomenon in the field of research. So you can imagine just one cubic centimeter material is divided into more than small pieces, then that can possess that high surface area. So high surface area can produce that some kinds of different properties of that material. So we think that there is some kinds of quantum effect dominate in that kinds of small size particle. So in case of gold, that is divided into the small size, just like a nanometer size, then that melting point has been changed, and also color or fluorescence and electrical property or magnetic property has been changed. So you can see here you can see here that 20 nanometer sized gold nanoparticle possess that the color has changed just like wine pink color, and bigger size possess that the purple colors. So in the nano means that under the micrometer size. So but normally 1 to 100 nanometer is that defined as a nano in NNI from US defined that kinds of dealing with 1 to 100 nanometer is for an industrial field. So related science, actually that is basic for chemistry and also physical, that inorganic chemistry, or sometimes we can apply it with molecular biology or other scientific field. But human race, earlier our human race adopt this technology into the kinds of Damascus blaze, and also that blaze contains that carbon nanotube, and also stained glass, that gold nanoparticle can show the different colors of the glasswares. So after almost 30 years devotion of the scientists of the nano field, they produce so many kinds of industrial products, such as quantum.TV or quantum something like a nano industry field. But they eagerly want to apply this nanotechnology to the medicine because of that huge surface area that can be applied with the multifunctional application, and also small modification that can change it into excretion or clearance in time, and also some nanoparticle has a porous structure that can we can deliver the drug inside of the nanoparticles. Up to now, I think in vitro or sensing is commonly used for nanomedicine, such as ultrasensitive nanosensing, also biomarker nanoimaging, or sometimes drug delivery vehicle, and then sometimes we can use the regenerative medicine. But we consider several factors for nanomedicine. You have to consider that the core synthesis. Sometimes we can make a hard core, or sometimes we can make a soft core nanomaterials, and also surface modification that can be applied for human use. And for radio labeling, we have to consider that the bifunctional chelating agent, and size and charge or shape will affect the biodistribution inside of the body. And also toxicity is a very important point for, especially for nanomedicine. Year of 2009, already that the size matter of the nanomaterials can differentiate the biodistribution. So up to 100 nanometer that can engulfed by the corpuscle in the liver, and less than 5 nanometer, and they are excreted via the kidney excretion. And also positively charged nanoparticle that can harmful for a cell, that can possess that cell toxicity. I think in nuclear medicine already we use the nano system for imaging, such as technetium HSA. They can visualize the GI bleeding, or nanocolloidal structure. They can help to find the lymph node or lymphatic systems. And radio-labeled nanomaterials that can help the whereabouts the nanoparticles, such as we can get the imaging or tissue-based biodistribution study. For the radio labeling part, I think we can think that simply bifunctional chelating agent we can use for the radio labeling. That is a easiest way to radio labeling. And surface labeling, that means the surface of the nanoparticle that can that radioisotope can attach to the surface of the nanomaterial, or sometimes we can mix with the core material with the radioisotope. So after the radio labeling, we can do the imaging or biodistribution study. Firstly, as I mentioned, that I'm a radiochemist to visualize that whereabouts the nanoparticles. So firstly, we set up the radio labeling method for each nanoparticles. So at more than 10 years back, we got several kinds of nanomaterial from our country, and then I attached the chelating agent on that, and then radio labeled it. But sometimes it's went well, and then sometimes it took more than 5 years to radio labeling set up. So I struggled for that kinds of radio labeling on the nanoparticles. So my mentor, Professor Jae-min Jung, proposed that, okay, that Yun-sang, you simply mix with the chelating agent or targeting molecule, and then you can get encapsulated nanoparticle. So we published the year of 2012. And you can see here, twin 60 or twin 80 is an amphiphilic molecule that can possess that hydrophobic chain, and also they can contain that packed molecule on one molecule. And then they mixed with the nanoparticle, we can get very stabilized or hydrophilized nanoparticle by ourselves. And after the radio labeling, you can see here that the tumor site, that nanoparticle distributed to the tumor site, and that is blocked with the specific molecules. So that is the all-in-one step functionalization kinds of things, and all the hydrophilization agent and functional the targeting molecule, and also chelating agent mixed with that can form a micelle, and then into the micelle solution that hydrophobic quantum dot or any kinds of hardcore materials drop into the that micelle solution, and then we can get hydrophilized nanoparticle. And after that, we evaporate the chloroform over hex and normal hexane, and then we can get hydrophilized nanoparticles. So that, firstly, we applied that thing to the quantum dot, and next one is the iron oxide nanoparticle, and we calculate the sugar moiety on the nanoparticles, and we can do because of that iron oxide nanoparticle can see the MR signal, so we can do the PET MR and PET study. As you can see here, after the hydrophilization, that nanoparticle possess that very monodispersed structure size, and you can see here after the hydrophilization encapsulation, just 10 nanometer size. And also under the high salt concentration, there is no change of the size, and after the radio labeling, there is no change of the size. So we apply that iron oxide nanoparticle to the lymph node imaging, in vivo lymph node imaging, that nanoparticle injected into the foot pad. And can you find any the darkening side of this image? But I couldn't find. But after get information of the pad part, and then we can magnify this area. And then finally, we could find that some particles aggregated in this portion. So that means we inject a small dose, almost 10,000 times less dose than the earlier ferridox or kinds of ML contrast agent. So we think that that radio isotope labeled nanoparticle nanomedicine can help reduce the toxicity of the imaging of the kinds of lymph node imaging. So we think that radio nanomedicine can help that nano better or radio better property of the nano product. And then after that, we radio labeling procedure did in on a nano inside of the nano just a solution inside the solution contains that the nanoparticle. So after that, we try to make a click chemistry on the nanoparticle. As you know that last year, click chemistry got the Nobel Prize in chemistry. Click chemistry is just a concept. That is not the specific chemistry route. Just simply easily formed and also easily separate from the starting material, and also even physiological pH in water that can be formed some kinds of molecules. So that is defined as a click chemistry. So firstly, copper was used for a catalyst for click chemistry, and also later on, copper-free click chemistry was developed by the Bartolgi, Professor Bartolgi, and then that can be used as a radio labeling procedure for all the peptide or nanoparticles. So we think that clickable nano platform can be applied for nanomedicine, such as targeting molecule decoration or just a radioisotope attached to the nanoparticles. Sometimes we can apply different kinds of radioisotope, such as a copper, lutetium. We can doubly labeled with one nanoparticles like that. And all set of the click chemistry on a nano platform, we focus on the nature invented nano platform such as albumin. I think an NM physician, you can understand what is the meaning of this image. This is the blood pool imaging agent, such as a technetium, just like a technetium HSA imaging, but somewhat different because of the after 4-hour injection, you can see the high blood pool activities, including carotid artery, and we think that that is somewhat different with the earlier technetium HSA imaging, because after the injection, within 1 hour, all the radioactivity go into the liver, even in a mouse and also human. So what is the difference? So we think that that click chemistry applied human serum albumin, that radio labeling procedure is outside of the albumin. But technetium HSA radio labeling procedure is so we use the highly reactive tin chloride. That tin chloride solution can meet the human serum albumin. So we think that that earlier technetium HSA is not the real meaning of human serum albumin. So we think that copper clickable human serum albumin can maintain the nature of the serum albumin. So that's why that blood circulation time is highly increased. As you know that almost 60% of protein in human blood, and also just a simple small modification can reduce the half-life. So after that, I got the patent of this conceptual study. Then after that clickable albumin nanoplatform can attach the radioisotope or targeting molecule or therapeutic drugs. And after that, we set up all the chemistry, and then we calculate how much molecule on the albumin nanoplatform, and then we can do the images with the diagnostic or therapeutic radioisotope. And firstly, we attach the folate for a targeting molecule. You can see the KB cell line that highly expressed of the folate receptor. So you can see here high uptake inside of the tumor, inside of this cell, and that is blocked with the folate. And after that, we got the in vivo imaging, and you can see here tumor inoculate the model. You can see the high uptake in the mouse model. And after that, okay, thank you. So after that, we attach the galactose and mannose molecule. That is the simply two position carbon and 4 position carbon contains that the hydroxyl molecule different. But one albumin can decorate the only 3 different carbohydrate. But you can see here that galactose labeled albumin can show only hepatocyte, but mannose-related albumin only show the corpuscle. So I found that in the textbook that our imaging and textbook is almost same. That this is the ex vivo imaging, so just simply mixed with the galactosyl albumin and mannose albumin into the IV injection, and then after getting liver tissue, and then we can see there is no staining or like that. So we apply this thing to the metastasis imaging, and then also applied to the technetium radio labeling method. And also we can apply I think today the day before yesterday, we discussed about the boron neutron capture therapy that can, that albumin nano platform can be applied for BNCT also. So my final goal would be already we set up all the kit formulation of these albumin nano platforms, and that can be used diagnose or therapy or theranostics for human use. So already I set up all the kit formulation, and monthly we produce more than 100 vials. And then if you want, I think maybe sooner or later, Dr. Nadim Lodi will get these kinds of kit formulation, and then he will apply this kit for new pharmaceutical production. Okay, thank you very much.
Thank you, Professor Lee, for your nice presentation. We know that nanoparticles have property to penetrate through the cell membrane. So the long biodistribution may be due to this cell penetration property, or there is inbuilt binding for nanoparticles.
Yeah, you are correct. But that depends on the nanoparticles, because of the cell penetration property is almost a 2D cultured cell line. But in case of 3D cultured cell structure, that tissue-based monitoring, that cell penetration property is dramatically decreased. So that means that is from the gravity of the because of the nanoparticle has a high density molecule structure. So I think yes, that not any kinds of nanoparticle can go to the liver, but sometimes we can see the lung. That means aggregated form of the nanoparticle. So I think we can discuss it later on after this session.
Okay.